Okay, so welcome back to this next video in which we are discussing ion channels and cancer. Okay, right, so in the previous videos what we've looked at is eferagogo 1 and eferagogo 2, which are both examples of voltage-gated potassium channels, okay, and which both seem to be proto-oncogenes potentially, okay, whereby if you get a gain-of-function mutation in them, okay, such as overexpression of them, that then leads to cancerous phenotype development. Okay, what we're going to do is continue now discussing ion channels that seem to have a proto-oncogene-like behaviour, a pro-growth function, and we're going to look at ion channels that seem to achieve this pro-growth function via interfering or regulating calcium signals. Okay, so calcium is one of the key intracellular signaling molecules. In fact, it's probably the second most important intracellular signaling molecule after potentially cyclic AMP. Okay, and calcium can regulate growth signals. It can regulate the growth of a cell as well as all the other things that calcium ions can do. Okay, right. So, let's have a look then at how an ion channel, specifically the intermediate conductance calcium-activated potassium channel, is involved in potentially potentiating the calcium signals generated by platelet-derived growth factor. Okay, so we're going to be discussing PDGF, which is a growth factor, platelet-derived growth factor. Okay, now growth factors are capable of inducing cells to divide. Okay, so they're capable of inducing cells to move from being in the G0 phase, the inactive phase of the cell cycle, to being in the G1 phase, the active phase of the cell cycle. So this is platelet-derived growth factor. Okay, right, so how does the platelet-derived growth factor act on a cell? Well, basically, cells have receptors for platelet-derived growth factor. So I'll draw my cell here, okay, and here is the receptor for platelet-derived growth factor. And basically, it's a receptor tyrosine kinase. So what happens is when the platelet-derived growth factor binds to the platelet-derived growth factor receptor, which is here in red, okay, what will happen is the platelet-derived growth factor will change conformation, okay, and then that new conformation that it forms will be capable of dimerizing with another platelet-derived growth factor receptor, which also has a platelet-derived growth factor molecule bound to it. Okay, so the change in conformation favors the dimerization of these platelet-derived growth factor receptors together. Okay, and then that platelet-derived growth factor receptor dimer is then capable of activating all sorts of downstream signaling pathways. Okay, so for instance, it can activate the RAS-RAF-MEK-ERK cascade. Okay, it can activate the PI3 kinase AKT-mTOR cascade. Okay, so it's capable of activating many pathways, which culminate in the nucleus ultimately, and they change gene expression generally. Okay, well, at least the RAS-RAF-MEK-ERK pathway culminates in the nucleus. Other pathways actually culminate by interfering with ribosomes, okay, so interfering with translation. Specifically, the PI3 kinase AKT-mTOR pathway interferes with translation, okay, but the idea is these pathways change gene expression, okay, change gene expression, and this is how you move from having a cell that's not dividing to having a cell that's beginning to divide, okay, so moving from G0 to G1. Okay, now, one of the signals that platelet-derived growth factor receptors are believed to produce is a calcium signal, which is believed to then also have effects on changing gene expression that help to induce the cell to divide, basically. So this can have effects that also help to change gene expression. Okay, now, the way that they are believed to do this is by opening plasma membrane calcium channels, which allow calcium to come in. Okay, now, there seems to be the involvement of another ion channel in potentiating this calcium signal, basically. Okay, indeed, what seems to happen is the other pathways that platelet-derived growth factor receptors induce change gene expression so that you upregulate the intermediate conductance calcium activated potassium channel. Okay, so one of the first genes that you now increase the expression of is this intermediate conductance calcium activated potassium channel. Okay, and I'll colour this one in turquoise here. And this is believed to then potentiate the calcium signal, because what's going to happen is, because of the presence of the high intracellular calcium, you're going to activate the opening of these intermediate conductance calcium activated potassium channels. Okay, that's then going to allow potassium to move out of the cell. Okay, and the idea is that this is going to counter the movement of positive charge into the cell in the form of the calcium. Okay, so by moving potassium out of the cell, you ensure that you're not depolarizing the electrical potential difference too much, that you won't be able to bring that much calcium in. Basically, if you open this channel, you're allowing potassium to move out. That's going to repolarize the electrical potential difference across the cell membrane, and that means that calcium's driving force to come into the cell is going to be strengthened, basically. So you're going to get more and more calcium coming in. Okay, so this is believed to play a key role in strengthening this calcium signal here. Okay, and that then is what, the calcium signal then has a pro-growth effect, and that's how this intermediate conductance calcium activated potassium channel is believed to potentiate the growth stimulation produced by the platelet-derived growth factor receptor when platelet-derived growth factor binds to it, okay, by potentiating the calcium signal in this way. Okay, right, so there's the involvement of the intermediate conductance calcium activated potassium channel in potentiating a calcium signal induced by the platelet-derived growth factor receptor. Okay, right, I'm going to mention now another example of a ion channel that works by interfering with calcium signals, which are then pro-growth, or at least that's believed to be how it has a oncogenic effect. Okay, so this example is a TRP channel now, so we're going to go to TRPM8. Okay, so remember the structure of TRP channels. TRP channels are tetramers. Okay, and each one of those subunits of a TRP channel has a similar membrane spanning topology to a voltage-gated potassium channel. Okay, so here is our TRPM8 tetramer. Okay, and it will be a homotetramer. All of these subunits will be TRPM8 subunits. Okay, so here is the TRPM8 channel. Okay, and it's a non-selective cation channel, so it allows in calcium, importantly, as well as sodium. Okay, right. So TRPM8 is quite a famous channel because of its importance in detecting noxious cold in sensory neurons, so pain neurons. Okay, indeed it responds to cold temperatures of around 17 degrees Celsius to 25 degrees Celsius. Now that might not sound that cold, but body temperature, remember, should be 37 degrees Celsius. Okay, so these are cold relative to body temperature. Okay, it also responds to the chemical menthol. Okay, which is why menthol has a cold and refreshing taste when it's in toothpaste. Okay, right. TRPM8 is not just found in sensory neurons, it's got a wider expression profile than that, okay? And there's certain examples of cancer cells where it's found to be hugely upregulated though, okay? So, particularly in prostate cancer. Okay, so prostate cells do usually express TRPM8. It's not quite as dramatic as EAG1, where it's only expressed in the CNS usually, and then you get the ectopic expression in cancer cells. No, TRPM8 is normally found in prostate cells. Okay, but in prostate cancer cells, you find much higher levels of TRPM8 than you would find in normal, healthy prostate cells. Okay, so the levels of TRPM8 are hugely increased in prostate cancer cells. Okay, and the reason that TRPM8 is hypothesized to have a role in prostate cancer is that when it opens, it can allow calcium into the cell, and it's believed that this calcium signal that potentially it allows into the prostate cancer cells is then pro-growth. Okay, so again, it's believed to have a pro-growth effect on these prostate cancer cells. Okay, and it is a very hopeful, promising target for future prostate cancer medications. Okay, right. Okay, so we'll call it there for this video, and in the next video what we'll do is discuss an example of a channel that isn't actually a pro-growth channel, but instead we believe is actually a tumour suppressor gene. Okay, i.e. it seems to have a role in stopping cells from over-dividing.